Okay, let's get started. So today we're going to talk about RNA-seq. We know that in the genome, we have microarray lectures now. Genes are transcribed into RNA, and these RNAs are then spliced with the introns spliced out, and at the end you will have a mature mRNA with introns out, and that's when it's translated is made into a protein. And we have microarray lectures to talk about how to use microarray to detect the expression of these RNAs. But today we're going to talk about RNA-seq. Basically, when you have the initial RNA, either mRNA or total RNA, you can remove the initial DNA from this, and then we shear the original RNA into smaller fragments. In fact, the technology for shearing this, not the shearing the RNA, but fragmenting the double-stranded DNA has been changing. Anyway, so the RNA is fragmented to about 200 to 300 base pair fragments. And then you use reverse transcription to create a double-stranded DNA, which represent the initial fragmented RNA. And these double-stranded cDNA can then be ligated to sequencing adapters. And these can then be PCR amplified and then put on high-throughput sequencing machine, as we mentioned in the last lecture, Illumina sequencing. And then we can do sequencing reads from either one end or both ends. So in this particular case, we are sequencing the cDNA from both ends. So each of the original fragments, you will get two read sequence, one from the plus strand, the other from the opposite strand. Okay, so this is the rough idea of RNA-seq. The protocol for this is still being improved, and the price of the library preparation is getting cheaper and cheaper. Why do we bother doing RNA-seq, not microarrays? Well, actually, it has a lot of advantages now. First of all, we do not need to know the genome sequence or even predicting genes. Remember, for microarray, in order to investigate some genome's RNA, you have to know the genome sequencing, you need to know the gene predictions and design specific probes against it. And so if we have a new, say we get an alien, and we don't even know their genome, right? But you do find out that they have DNA or RNA. And so one simple way is just to get the RNA and sequence it out without even knowing the genome sequence is okay. Yeah, so we do not need to design microarray probes in here. And it's a digital representation. Every time you read out a fragment, you know this is one of the original transcript coming from the genes. Whereas for microarray, it's a hybridization, kind of an analog average signal of many transcripts aligning to the same sequence. And RNA-seq have higher dynamic range. So for microarray, eventually the color for the fluorescence will plateau for the highly expressed genes, and also at a low level, you may not really see the signal strong enough. Whereas RNA-seq, every time you read out a sequence, you know that this is coming from the original RNA. We can also use it to identify new genes. If you didn't, like this alien, you didn't even know what genes they have. So you can use RNA-seq, and everything you see transcribed is probably coming from a gene. You can also use it to look at alternative splicing, which is, for example, we mentioned that for a particular gene, you could have exons and introns and exons and introns. But from the same gene, there could be multiple transcripts using different combination of the exons. So the exons are the ones that are eventually going to be productive in terms of making a real transcript going to protein translation. But different transcripts might use a different set of the exon. For example, one transcript might just use exon 1, 3, and 4. The other will only use 1, 2, and 3. Towards the end of microarray days, Affymetrix did try to use array to look at alternative splicing, but the signal was very bad, so it was never really popular. But with RNA sequencing, you can now really look at splicing. We can also see mutations on the gene. based on the RNA, and also because you read out every single base on that gene. So if there's mutation, you can see it. Or gene fusion, which is one gene fused with another gene, and you can read it through and see how somehow this RNA read go from one gene to another gene, then you can see it. Okay, so it has a lot of very, very good applications in here. In terms of experimental design, there are many ways you can do RNA, because within the nucleus, when the gene is just transcribed, you get both the introns and the exons. But once the RNA become a mature RNA, after splicing, it will go to the cytoplasm for translation. And so there you only get the exons without the introns. And so there is a technique using poly-A or using poly-T to basically only enrich for the mature mRNA transcript. There you get a better signal. But sometimes people want to know the intronic sequence as well. They just need to get rid of some very simple like ribosomal housekeeping genes. So these are just too abundant. You will be wasting a lot of your sequencing reads on the genes that are really just doesn't really matter for, well, it's important, but biologically, it probably is not interesting for research. And so you can use something, a bead, kind of to remove all the ribosomal RNA. This way you will enrich for the real other genes in the genome. People are also trying to look at the strand specific. You know, sometimes you want to know, because after you get the cDNA, you will sequence the two ends. Sometimes you don't know which strand it is coming from. So depending on the different protocol, you will get some slightly different things. Most of the people just use standard poly-A mRNA splicing, especially if you're dealing with human samples or things like that. It's just simpler. And there are sequencing facilities now, and the cost of sequencing, 10 years ago, it's about $2,000 to $3,000. Nowadays, you can, and that was only sequencing cost at the time. The reagent cost was extra. And nowadays, you can just even get a piece of tissue. Some of the facility ask you to isolate RNA, and some of them say, well, just send us, you know, whatever tissue. They can help you isolate RNA, make a library for putting on a sequencing machine, and sequencing a sample for $300. It's like amazing. Some of the facilities is even offering library preparation and sequencing for $199, which is really great. However, as a scientist, especially as a computational scientist, you should understand what does $300 a sample mean. For example, when you do the sequencing, you could sequence from either one end or the other end. In terms of the sequencing time, sequencing both ends will take twice as long as sequencing one end, and you will get twice as much data, right? So usually single ends theoretically should be cheaper, but recently for a lot of these sequencing machines, they just default, since they are sequencing so many samples together, they're gonna sequence paired end together. In terms of sequencing depth, for example, you can imagine you have this DNA. Right now, with one sequencing that flow cell, you can put 5 samples there. You can put 10 samples there. You can multiplex even 20 samples in there. And so each sample will get either 10 million reads, 30 million reads, or 500 million reads. And so when you say sequencing is now $300 per sample, we have to ask them, how many reads am I going to get, right? And also, if it's say 30 million reads for a paired end sequencing, that is 15 million fragments, because you get 15 million reads on the left side of the, so in the sequencing, like here, you get 50 million reads on one end of the transcript or of each fragment, and then 50 million reads for the other end of each fragment, right? So when you look at the sequencing depths, depending on whether it's single end or paired end, you also have to ask them exactly how many fragments you're gonna get and how many reads you're gonna get. For example, if you only want to look at expression level and calculate differential expression, probably 20, 30 million, or yeah, 50 million should be definitely sufficient. However, if you want to look at, say this is alien genome, we don't even know its gene, right? It's a lot harder. There for like transcript assembly to figure out which genes are going to transcribe and look at splicing or alternative splicing, you probably want to go deeper, like 200 million reads. Read length is another factor. So initially, when sequencing machines were invented, the machine can only reliably read out about 25 nucleotide. If you run fast QC, you can see the quality of the bases decrease as you read longer. But as the technology improved over the last 10 years now, we can very reliably get 150 base pair reads now. But again, for the same machine, if they sequence, remember every cycle of the reaction, you sequence like A, C, T, G, and then you add another A, C, G, T. And so if you have to run this machine to sequence 50 base pair, from the same machine, if you have to sequence 150 base pair, that 150 base pair will take 3 times longer on that machine. And so the sequencing facility should charge you more if they sequence deeper, or if they sequence more reads, or if they sequence longer, right? So the price, don't be fooled just by the price. You need to know, am I getting, how many reads am I getting? How long are the reads, right? That's actually very important in the pricing factors. Nowadays, which the amazing thing is in facilities, even paired end sequencing, each end 150 base pair long, and at a 20 million fragment, you can get $200 per sample. And at this price, it's definitely cheaper than microarrays. When RNA-seq was just started, biologists really believe that the advantage is that we get real digital signal. Every time you read out something, it is a real transcript coming from the original sample. So the noise should be lower, which is true, but they feel like we didn't have to do replicates. Now that well, at that time, sample sequencing was just too expensive. You want to do the minimum or spend this amount of money to get your studies published. And so at that time, they thought once, you don't need to replicate, one sample is enough. But really, in order to really assess biological variation, you still need biological replicates. For example, if we want to know what is the difference between a disease versus normal human, there are a lot of variabilities just between individuals. So there, you probably won't be able to save much. For a technical experiment like cells growing on a petri dish, now mostly people do 2 replicates. This 2 will get you enough for publication. 1 is only for exploratory assays. You just want to see, oh, if I tweak the cell, does it even work at all, right? You generate a single replicate, and just to quickly look at the result. But in order to publish, 2 is kind of the minimum, 3 is still preferred. Batch effect still definitely exists. As we mentioned, you can think of batch effect just like a biological treatment. Because of that technician is sloppy or something, and all the samples, their hands could influence the experimental condition in some way, right? So batch effect definitely still exists. So in terms of experimental design, try to be consistent and process all the samples together in as few batches as possible. And also try to avoid the complete confounding effect, which is if all the normal samples are processed in one batch and all the cancer or like the other condition is processed at another time, try to eliminate those situations. Yeah, so better technology never eliminate the needs for good experimental design, okay? For one example of that is there is a striking finding in 2014 published in PNAS, which says, human heart is more similar with human brain than mouse brain. And so basically, in this experiment, the study basically took different tissues from human and mouse. You can see here are the samples. You see the the colors indicate the tissues, and the shape indicates whether the samples are coming from human or mouse. And you can see here, all the human samples are here, and all the mouse samples are here, which, yeah, pretty much means human brain is closer to the human heart than the mouse brain. which if it is true, it would mean that all the previous study done in model organism is not worth looking at. Right? So you think about this, ideally we want to do all the experiment in human to understand human biology, but the reason it's not feasible is a lot of times you can't really experiment on human. That's why people use yeast, they use worm, they use fly, they use mouse as a model organism to study human biology, hoping that some genes, if they function the same way in the mouse brain, they will function in a similar way in human brain. But if this is true, then all the previous model organism study would be invalid. So a few months later, another scientist published a study to show that, say, we reanalyzed the data from this study, and they found that the first batch is all human tissues, second batch is all human, the third batch is all mouse, the fourth batch are mouse tissues, and so on. But after they do the batch removal, they do see that the same tissues, whether it's in human or mouse, are now clustered together. So we have to be very careful about this, okay? Yeah, question, Jed.
Was that study retracted?
I don't think so. No. Right.